Hello everyone. I hope all of you are doing well. Welcome to Vinay Kumar Tutorials. From this channel, we are making an attempt to deal with various concepts related to life sciences and biology that will help the learners who are studying in life sciences stream in various courses and also who are preparing for competitive examinations. So let us start with an interesting topic, that is concept of origin of life. The contents that we are studying in this topic are introduction, theories of origin of life, process of origin of life, evolution of progenote, evolution of eukaryotes, current research, and conclusion. So now let us start with introduction to the topic. The question of origin of life on our planet, the Earth, has remained a most complicated and puzzling problem for thinkers, philosophers, and naturalists of modern as well as ancient times. Different views have been forwarded concerning the origin of life on Earth by thinkers of different ages. It is generally agreed that all life today evolved by common descent from a single primitive form. It is agreed that all the life form which are today, they arrived and they existed and evolved from a common descent, that means a common ancestor. We do not know how this early form came about, but scientists think that it was a natural process which took place perhaps 3900 billion years ago. This concept is in accordance with a philosophy called naturalism. Here only the natural causes are admitted. First sign of life on Earth. Earth is estimated to be about 4.5 billion years old, and for much of that history, it has been home to life in one weird form or another. Indeed, some scientists think that life appeared the moment our planet's environment was stable enough to support it. Studies of genetic material indicate that a living group of single celled organisms called archaea may share many features with early life on Earth. The oldest evidence for life on Earth comes from fossilized mats of cyanobacteria called stromatolites in Australia. That are about 3.4 billion years old. This is very important. The oldest recorded evidence of life are stromatolites. These are mats of cyanobacteria which are located in Australia, and according to a report, they are existed about 3.4 billion years old. Theories of origin of life. There are numerous theories which are presented regarding origin of life. The important theories are given as follows. First one is special creation theory, next religious beliefs and hypothesis, abiogenesis theory, biogenesis theory, hypothesis of panspermia, theory of chemical evolution, proteinoid microspheres model. The first and foremost theory is special creation theory. Life is originated from supernatural event by supernatural power, that is God. It cannot be studied scientifically but must be accepted on faith. According to this theory, life is created by God, and the theory emphasizes that this theory cannot be studied with logics or scientific basis, but it must be accepted on faith. Second theory is religious belief and hypothesis. According to Hindu concept, Lord Brahma created the living world in one stroke. Christian and Islam belief says that God created this universe, plants and animals in about 6 days. The three main postulates of this theory are: all different plants and animals are created at once. All organisms were created in the same form in which they exist today. Their bodies and organs have been designed to meet the needs of the environment, but it has no scientific basis. Next, abiogenesis or theory of spontaneous generation. The name or the term abiogenesis itself indicates that A means absence, bio means life, genesis means formation. The formation of life from non living substances is called abiogenesis or spontaneous generation. The belief in abiogenesis started from 300 BC and continued until 17th century. Aristotle and Epicurus believed that fishes originated from mud, insects from decaying meat and worms, and many other animals originated from soil and manure by the action of wind and rain. Louis Pasteur disproved this theory and gave the scientific explanation that life originated from pre existing life or biogenesis. Abiogenesis theory focuses upon the formation of life from inanimate matter, that is, non living thing. But later, the series of experiments which were conducted by different biologists, including Louis Pasteur, disproved the theory of abiogenesis and gave scientific explanation about life which is existing from pre-existing life. The next theory is biogenesis. Biogenesis theory refers to origin of life from pre-existing life by reproduction. The supporters of this theory are Francisco Redi, Spallanzani, and Louis Pasteur. First of all, Francisco Redi conducted a simple experiment to support the theory of biogenesis. The experiment which he conducted was very simple. He took three flasks and kept cooked meat in all the three flasks. The first jar or the flask was uncovered, that means it was kept open. The second flask was covered with a rubber cork, it was sealed, and the next third flask was covered with gauze. After a few hours, he observed that the flies entered into the first jar easily, and the flies fed on the meat and they laid the eggs. Later, the eggs hatched and the larvae or the maggots appeared on the meat. In the second condition, that means in the second flask, there was no entry of the flies into the flask. Why? Because the flask was tightly sealed. But in the third condition, as the flask was covered with gas, the flies could not enter into the flask, but they remained on the flask. They came near the flask. In the third condition I am talking about, in the third condition or in the third flask, the flies came near the flask only because of the smell, and they settled on the gas only. So by this simple experiment, Francisco Redi concluded that the larvae or the maggots which are appeared in the first flask, it is because of the flies that has flies that have entered into the flask, and in the second and third flask we have not seen any life. Why? Because the flies failed to enter into the second and third flask. So at last Redi concluded that there should be some pre-existing life which can give rise to a new life. But so many biologists and other naturalists did not accepted his theory completely. So Lazzaro Spallanzani conducted two experiments. Here, Spallanzani took broth. Broth means a liquid medium which contains nutrients. Generally, broth is used to culture the microorganisms as a nutrient media. So here he Spallanzani took a broth in the round bottom flask and he heated the broth. Then he cooled the flask. Then the flask was left open. He waited for few hours. Later, when the broth was tested, it showed the presence of microorganisms. In the next experiment, he took the broth again and heated. Next, the flask was sealed tightly. As the flask was sealed tightly, there was no chance for air or spores to enter into the round bottom flask. After breaking up the neck of the flask and then after testing the broth or the contents, the content showed the presence of microorganisms. So in the first case, as the flask was left open, what happened? there was chance of air. Along with air, some spores of fungi or other microorganisms may have entered into the flask. That's why the broth showed the presence of microorganisms. In the second case, as the flask was sealed, and then the contents which are present in the sealed flask were immediately tested, the broth did not show the presence of any microorganism. But when the same flask was left open for few hours later, when the contents were tested, it showed the presence of some life forms. So next, Louis Pasteur conducted experiments which ruled out the theory of abiogenesis. That means it completely disproved the theory of abiogenesis or spontaneous generation, which was the experiment that was conducted by Louis Pasteur. Louis Pasteur conducted few experiments which clearly disproved the theory of abiogenesis. He took the broth and boiled the broth in the round bottom flask. The end of the neck of the round bottom flask was designed like the swan's neck, and the end of the tube was completely sealed. When the broth was boiled and the apparatus was kept undisturbed for a few hours. After a few hours, when the contents present in the round bottom flask were tested, it did not show the presence of any microorganism. In the next case, or in the next experiment, Louis Pasteur took the same flask and boiled the broth in that, and then the neck of the round bottom flask was broken and it was kept for few hours. Next, after few hours or after some duration of time, when the broth was tested, it showed the presence of some life forms. Here, the air along with the spores which entered into the round bottom flask is responsible to create the life. That means the spores, which are also microorganisms, because air has so many microorganisms. Microorganisms are present everywhere. The microorganisms which are living forms, they have entered into the flask through air as a medium, and then they replicated or reproduced in the broth. Using that broth as a nutrient medium, they replicated or reproduced. And when at last, when the broth was tested, it showed the presence of different life forms. So by this experiment, Louis Pasteur disproved the theory of spontaneous generation and concluded that life originates from pre-existing life. Next theory is the theory of panspermia. It is also called cosmozoic theory and was developed by Richter in 1865 and then supported by Thomson Helmholtz in 1884. According to this hypothesis, meteorites traveling through the Earth's atmosphere are strongly incandescent only on the surface while the interior remains cold. Thus, the embryos of organisms inhabiting meteorites or the planets from which they were formed were preserved in the meteorites, finding fertile soil on Earth and grew and evolved to produce all the new species. So, the main hypothesis of panspermia theory is the life came from extraterrestrial spores transported by meteorites. As this theory as this theory is not having any scientific evidence, this theory was rejected. Next comes the important theory of chemical evolution. The supporters of this theory were Alexander Oparin and J.B.S. Halden. The conditions on the primeval Earth billions of years ago were different from present day conditions. The first life or the self-duplicating particles did arise spontaneously from chemical inanimate or abiotic substances such as proteins, nucleic acids, etc. The primordial atmosphere of Earth had water, methane and ammonia from which, as a result of series of changes, a colloidal body called coacervate was formed. The coacervates contained a mixture of biologically important macromolecules like proteins, lipids, and nucleic acids. These molecules were not living, behaved in a manner similar to biological systems by being subjected to natural selection, by being chemically directive and having the capacity of reproduction by fragmentation. Theory of chemical evolution stressed on coacervates. What are these coacervates? These coacervates are the particles which contain mixture of macromolecules like proteins, lipids, and nucleic acids. It has been assumed that these coacervates were the precursors of synthesis of the genetic material and ultimately for the generation of life. The supporters of this theory argued that the Earth's prebiotic oceans, which were very different from present oceans, would have formed a hot dilute soup. In this soup, organic compounds, the building blocks of life, could have formed, and this idea was called biopoiesis. Lederberg has recognized the following three types in the Oparin's thesis. First one is chemogeny, next biogeny, and the third one is cognogeny. Chemogeny indicates chemical evolution. Biogeny indicates formation of self-reproducing biological units. Cognogeny refers to the evolution of mechanisms of expressions, communications, perception, and feeding. Oparin and Haldane got the ideas from the studies of biochemistry made by F.G. Hopkins, and both were of the opinion that evolution is purely chemical, the gradual transmutation of inorganic compounds into organic ones. So you can see the pictures of Alexander Oparin and J.B.S. Haldane in this slide. So next, the experimental support of Oparin's hypothesis. Stanley Lloyd Miller conducted experiment to support the chemical theory of evolution. It is generally called Miller-Urey or Urey-Miller experiment. Stanley Lloyd Miller was a Jewish American chemist who made landmark experiments. Miller synthesized organic compounds under conditions resembling the primitive atmosphere. In this experiment, a mixture of gas containing water vapor, methane, hydrogen, and ammonia was circulated through a closed apparatus by steam from boiling water. This is subjected to an electric spark discharge between tungsten electrodes. The electric discharge duplicated the effects of violent electrical storms in the primitive universe. Here, Urey Miller took some of the gases which were present in the primitive atmosphere. It was assumed that water vapor, methane, hydrogen, and ammonia were present in the primitive atmosphere. So, Miller collected these gases, and these gases were circulated through a closed apparatus by steam which was taken from the boiling water. These gases were again subjected to electric spark discharge between tungsten electrodes. Why it was done? Because it was assumed that the primitive atmosphere was containing lightning with heavy thunderstorms. To recreate that atmosphere in the lab conditions, Urey Miller subjected those gases to electric spark discharge between tungsten electrodes. The electric discharge duplicated the effects of violent electrical storms in the primitive universe. The gas mixture was then condensed and added to the boiling water for recirculation, and the apparatus was permitted to run for a week. Later, several amino acids were synthesized. Example, alanine, glycine, valine, serine, and a number of other organic compounds such as hydrogen cyanide, aldehyde, and cyano compound, and small molecules that make up proteins and carbohydrates. This was the Urey-Miller experiment. Next, proteinoid microspheres. In 1964, Sidney W. Fox reasoned that proteins were synthesized from amino acids in the primitive earth by thermal energy or heat. Fox heated a mixture of 18 amino acids to temperatures of 160 to 200 Celsius for varying periods of time. He obtained stable protein-like macromolecules which he termed proteinoids. Later he observed small spherical cell-like units that has arisen from aggregations of the proteinoids. These molecular aggregates were called proteinoid microspheres. Physical properties. What are the physical properties of these proteinoid microspheres? Spherical, microscopic and of uniform diameter. Size in between the range of 0.5 micron to 0.6 microns. Properties in common with coccoid bacteria and microfossils found in ancient geological strata. The structural properties: ability of motility growth, capable of binary fission budding fragmentation, found to have catalytic activity such as degradation of glucose. Significance of proteinoid microsphere model. Proteinoid microspheres arise from monomers rather than from polymers obtained from organisms already in biota, as is true for the usual experiments with coadjuvate droplets. This model suggests that proteinoids are informational and it shows the origin of communication which may be intercellular or intergenerational communication. So, according to proteinoid microsphere model, these proteinoids they were able to communicate in between the two cells or in between the two generations. Darwin's views on origin of life. Darwin believed the possibility of natural emergence of the first life. He rejected the idea that purification of pre-existing organic compounds could lead to the appearance of organisms. In his book Origin of Species, he has mentioned that the primitive earth consisted of a warm little pond with all sort of ammonia and phosphoric acid salts, light, heat, electricity present, that a protein compound was chemically formed. These compounds undergone more complex changes and gave rise to life. The first genetic material. Since both proteins and nucleic acids are required to develop and reproduce organisms living today, an obvious question is which of these substances arose first, whether it is a protein or nucleic acid. Cairns-Smith's model. The first genes were minerals, minute crystals associated with protein as concentration sites and to some extent as templates. Certain minerals, example clay or other layered silicates, were in very large supply as products of early rock weathered. They might have favored the formation of classes of polymers for which they provided good templates. Natural selection would favor a polymer whose activities preserved or even increased the supply of its mineral template, perhaps by helping to weather a parent material to produce the clay gene. The association of two different polymers might have aided in mineral formation to provide building block materials for the colony, and polymer in each colony would have proteins. Eventually, a nucleic acid was added to a colony, and this nucleic acid replaced minerals as genetic materials. Presumably, the nucleic acid gradually took over the task of coding for all the proteins in the colony, including those associated with its own replication. Eventually, DNA evolved, and at some stage, membranes were developed around the colonies, and those membrane bound units became cells. This model was highly speculative, but evidence are lacking to prove the origin. RNA first model. In recent years, evidences are in favor of RNA to be material of first formed gene. A number of protein-free, that is non-enzymatic reactions that could have been decisive in the development of first biologically important polymers have been discovered by molecular biologists. The reactions include formation of RNA oligonucleotide from activated monomeric precursors, template-directed RNA synthesis, site-specific RNA cleavage, RNA-RNA splicing. Thus, RNA may have been the first polymer, and some form of reverse transcription may have given rise later to DNA. Process of origin of life. The process of origin of life can be studied in the following stages. First one is structure of cosmos. The universe or cosmos is made up of countless aggregation of stars organized into systems of various shapes and types called galaxies. These galaxies, each of which includes hundreds billions of stars, with their accompanying planets and satellites, are distributed into the vast reaches of space Milky Way galaxy. It is composed of 100 billion stars besides sun and its planets, that is solar system. The sun is a star being 5 billion years old and remains in a rapid state of motion. The 8 planets of solar system move, rotate around their axis and revolve around the sun in elliptical paths determined by gravitational force. Totally, the structure of cosmos gives us an idea of about the dimensions of the solar system. Primitive earth. Primitive means what were the conditions which were there in the ancient earth. That means when the earth was formed after the Big Bang theory, later the conditions on the earth were different compared to present day conditions. How was the primitive earth, first formed earth? Primitive refers to first formed. What was the condition of first formed earth? It is a tenable hypothesis that the cosmos originated from the gigantic explosion, Big Bang, which blew matter from a primordial fireball, scattering outwards the materials of the universe in an ever expanding volume. Clouds of dust and gases, chiefly helium and hydrogen, spread through the space. Later, the stars formed from the condensation of such clouds, and our own sun formed from the rotating mass of dust and gases. At some distances from the protosun, matter would be condensed into separate bodies. These would be the planets which lie in the plane of the sun's rotation. The solar satellites, including the Earth and presumably the other planets and asteroids, and the Earth's moon as well, appear to date from about 4.6 billion years. So here is a picture which represents our Earth 4 billion years ago. You can study the components along with the conditions in which they were present through this picture. Prebiotic synthesis. The core of the primitive earth melted from heat of condensation, but the outer crust cooled quickly and the primitive atmosphere was formed as various gases escaped from the interior of the earth, that is outgassing phenomena. The early gas cloud was rich in hydrogen. The hydrogen of the primitive earth chemically united with carbon to form methane, with nitrogen to form ammonia, and with oxygen to form water vapor. The crust and atmosphere of the primitive earth cooled slowly, and the first shallow seas were formed as the less volatile gases, including water, rained back down to earth. The oceans were formed as a result of millions of years of torrential rains, and the stage was set for the combination of the varied chemical elements. Chemicals from the atmosphere mixed and reacted with those in the waters to form a wealth of hydrocarbons. Water, hydrocarbons, and ammonia are the raw materials of amino acids, which in turn are the building blocks for the larger protein molecules. Finally, in the primitive seas, amino acids accumulated in considerable quantities and became kinked together or linked together to form proteins or oligopeptides. Primitive C served as a broth for the prebiotic synthesis. Classical experiments of Miller in 1953 has established that organic compounds can be formed without the intervention of living organisms. Thus, it appears likely that the sea of the primitive earth spontaneously accumulated a rich mixture of organic molecules. The sea became a sort of dilute organic soup or broth, and in this organic soup, the molecules collided and reacted to form new molecules of increasing levels of complexity. Proteins capable of self-replication must also have developed. Evolution of Progenote. The pre-cellular evolution involved the following six main steps. First is origin and evolution of RNA world. The machinery for self-replication was evolved before the development of the metabolic or enzymatic machinery of the cell, and it is likely that self-replicating polynucleotides slowly became established in the primordial earth some 3.5 billion years ago. Highlights of RNA world include distinction between genomic and functional RNA molecules, recruitment of exons into transcription, inclusion of introns in most genes, no regular cell growth. Second, origin of ribonucleoprotein world, RNP world. Certain polynucleotides direct the synthesis of macromolecules entirely of different types, namely polypeptides, proteins. Highlights of RNP world include origin of primitive tRNA, rRNA, and amino acyl tRNA synthetase enzyme, evolution of template dependent translation apparatus, origin of plasma membrane. A selective advantageous RNA molecule would be one that directs the synthesis of a protein that accelerates the replication of that particular RNA. The free diffusion of proteins could be prevented is some form of compartment evolved to enclose the specific protein made by a particular RNA. Most probably, the first cell was formed when polarized films of phospholipids forms soap-like bubbles enclosing aggregations of complex macromolecules. DNA world, the fourth one. At some later stage in the evolutionary process, DNA took the place of RNA as the repository of the genetic information, and this step may have occurred due to evolution of an enzyme called inverse transcriptase. Evolutionary highlights of DNA world include evolution of multiple pieces of DNA, stable storage of information in DNA, sure existence of plasma membrane, growth and division. Next, fifth one is origin of progenote. The precellular stage of organization has been termed as progenote. The progenote might have been a functional, slow replicating, independently evolving organism contained DNA, most genes with introns, a slow growth and heterotrophic nutrition. The primitive one-celled heterotroph probably had little more than a few genes, a few proteins and a limiting plasma membrane. Eventually, living systems evolved the ability to synthesize their own organic requirements from simple inorganic substances. Thus, in the course of time, autotrophs arose. Primitive autotrophs would require a whole array of enzymes to direct a multiple chain of metabolic reactions involved in the synthesis of a protein. The early fermentative autotrophs were much like our present day anaerobic bacteria, such as lactobacillus species and yeast. The metabolic processes of the anaerobic autotrophs resulted in the liberation of large amount of carbon dioxide into the atmosphere. Once this occurred, the way was paved for the evolution of organisms that could use carbon dioxide as the source of carbon in synthesizing organic compounds and could use sunlight as the only source of energy. Such organisms would be the photosynthetic cells. Early photosynthetic cells probably split hydrogen containing compounds such as hydrogen sulfide. The hydrogen is used by the cell to synthesize organic compounds and the sulfur is released as a waste product. In time, the process of photosynthesis was refined to the water served as the source of hydrogen and the result was the release of oxygen. At this stage, free oxygen became established for the first time in the atmosphere. The first organisms to use water as the source of hydrogen in photosynthesis were blue-green algae, that is cyanobacterium. Since blue-green algae were active photosynthesizers, atmospheric oxygen accumulated in increasing amounts, reducing primitive atmosphere into oxidizing type. Many primitive anaerobic bacteria incapable of adapting to free oxygen remained in portions of environment that were anaerobic, such as sulfur springs and oxygen-free muds. Geologic evidence indicates that free oxygen began to accumulate in the atmosphere about 2 billion years ago. The rising levels of atmospheric oxygen set the stage for the appearance of one-celled eukaryotic organisms, which arose at least 1 billion years ago. Later, within the comparatively short span of last 600 million years, the one-celled eukaryotes evolved in various directions to give rise wealth of or diversified multicellular life forms inhabiting the earth. Evolution of eukaryotes. The simplest cells in nature, the bacteria and blue-green algae, cyanobacteria, have been classified as prokaryotic cells. These cells have no nuclear membrane and lack specialized cytoplasmic bodies such as mitochondria and chloroplasts. Phylogenetically, the progenote evolved into three types of cells: archaebacteria, eubacteria, and eukaryotes. Archaebacteria are ancient bacteria. These are prokaryotes which appear to have diverged from the true bacteria, that is eubacteria, very early in evolution. The strange class of archaebacteria consists of
thermophiles, halophiles, methanogens. Next one is eubacteria. Includes cyanobacteria, chloroxybacteria, non-sulfur bacteria, green filamentous bacteria, and spirochetes. Eukaryotes. The cells consist of distinct nuclear membrane that encloses strands of DNA and consists of membrane-bound cytoplasmic organelles. In the eukaryotic plant cell, a prominent organelle is the chloroplast, the photosynthesizing particle that converts light energy into chemical energy. Endosymbiotic hypothesis. Evolutionary theory that explains the origin of eukaryotic cells from prokaryotic cells is endosymbiotic hypothesis. It states that several key organelles of eukaryotes originated as a symbiosis between separate single-celled organisms. According to this theory, mitochondria, chloroplasts, and possibly other organelles representing formerly free-living bacteria, that is prokaryotes, were taken inside another cell as an endosymbiont around 1.5 billion years ago. Molecular and biochemical evidence suggests that mitochondria developed from proteobacteria, rickettsials, and chloroplasts from cyanobacteria, nitrogen-fixing filamentous bacteria. Invagination of surface membrane hypothesis. According to some biologists, the evolution of living organisms normally proceeds through a series of gradual changes rather than the symbiosis. The continual modification of the surface membrane may have been the basic evolutionary mechanism in the differentiation of eukaryotic cell from prokaryotic cell. The organelles of eukaryotic cells may have evolved by the invagination, drawing inward of the surface membrane of a primitive cell. It is believed that many specialized internal structures of eukaryotes, including the membrane enclosed nucleus, may have differentiated from invaginated cell surface. So, current research. Big Bang experiment conducted at the European Center of Nuclear Research in Geneva focused on the concept of origin of life. What are the outcomes of the experiment? They discovered the hypothetical Higgs boson particle, also called God particle, which led to the existence of life. And so many researches to show close linkage between the physical properties of amino acids, the genetic code, and protein folding was likely the key factor in the process of origin of life. According to my opinion, the origin of life is still an uncleared concept, and it has been discussed from many biologists from the time immemorial. Many things have not been discovered about the origin of life. This point of view, none of the theories could be strongly proven, and it is presumed that over a few 100 million years of evolution, prebiotic molecules evolved into self-replicating molecules by natural selection. Several kinds of investigations are still being carried out, maybe one or the other day. The truth may be revealed, but until the mystery remains unsolved. And lastly, one important thing, if you want me to discuss the topic of your choice related to biology or life sciences, please let me know in the comment section below. So if you find this video helpful, please like, share and subscribe it, and press the bell icon below so that you will get the notifications when I upload a new video. Keep watching, keep gaining and keep smiling. Thank you.